As we begin to navigate through the different metabolic pathways, you should begin to recognize that as there are so many different pathways that intertwine with each other, this is an incredibly complex topic. It is so complex that we must regulate these different pathways. So now what we're going to do is discuss some of the generalities we can come to when we think about ways that metabolic pathways can be regulated, and then we're going to go into some detail on how glycolysis, gluconeogenesis are regulated, since those are the pathways we have already discussed. When we're thinking about the regulation of our different pathways, we need to make sure we review the term homeostasis. As you've learned, I'm sure in many of your other classes, homeostasis is the organism trying to get to the situation that it likes and maintaining that situation. And use the word situation because we're not always talking about metabolism. In terms of metabolism, homeostasis means we're trying to keep all of our metabolites at a steady state. This does not mean we are all at equilibrium. If at any point all of our metabolic pathways were at equilibrium and there was absolutely no change, we would probably be dead at that point. When we think about our different pathways, we want each of our metabolites to have a rate of synthesis that equals the rate of breakdown. Now, this is in a perfect situation when there's no environmental factors involved. When something in the environment changes, that is when the pathways are going to move in one way or another, always attempting to get back to that homeostasis. When we think about regulation, regulation is always our attempt to maintain homeostasis. If we have some sort of event that occurs that pushes the pathway in one direction or another, we will need to regulate different enzymes so that we are moving in the direction that we need to get back towards homeostasis. Regulation is incredibly important in our lives. A lot of the diseases that you're going to discuss as you continue your education, you will figure out stem from a lack of regulation or improperly regulated enzymes within the metabolic pathways. The most common type of regulation we're going to see as we move through the pathways is feedback inhibition. Feedback inhibition is very simply when the product goes back to inhibit one of the initial enzymes in a pathway. Since we are going to be discussing glycolysis and gluconeogenesis, we can go ahead and think about the ultimate goal of glycolysis as it moves through pyruvate into acetyl-CoA and into the citric acid cycle. The ultimate product is ATP. Therefore, we're going to see ATP go ahead and inhibit the enzymes that are all the way back in the beginning of our glycolytic pathway. Other than feedback inhibition, if we are attempting to regulate, there's a couple different ways that we can do this. We can work with the concentration of reactants and products, and we saw this quite a bit when we were going through glycolysis. If, for example, we have a reaction that is not spontaneous, it may have a positive delta G, what we can do is constantly remove the product from the situation. Removing the product is going to allow us to pull the reaction more towards production of product. What we're going to spend time with in this chapter is looking at how we can affect the activity of the catalyst by changing the concentration of the enzyme or the overall activity of the enzyme. We will also discuss how we can change the concentration of different effectors. And what I mean by that is a competing substrate, or maybe something like citrate, which can act as an allosteric regulator of the enzyme. Now, temperature can have an effect on biochemical reactions. We learned back several weeks ago that by increasing the temperature, we can increase the activity of an enzyme. But when we're thinking about the human body, temperature is not really something that we're going to deal with because we have a pretty sustained temperature. So this picture kind of gives you 10 different ways that we can affect the activity of an enzyme. Number one, we can use extracellular signals And we saw this some as we have discussed the effect of insulin and how it can bind to a tyrosine kinase receptor and things of that nature. We can see an alteration in transcription of genes. Some of our extracellular signals are actually responsible for going into the nucleus and affecting whether or not those genes are transcribed. We can see mRNA degradation if we degrade the mRNA, or if we inhibit the mRNA translation on the ribosome. That can decrease the amount of protein and hence the amount of enzyme we produce. The protein or enzyme itself can be degraded or sequestered into some sort of organelle, such as within the endoplasmic reticulum where it doesn't have any activity. We can affect an enzyme by, of course, binding the substrate. That's going to make it do what we want it to do. We can affect the enzyme by having some sort of ligand that binds somewhere other than the active site, which has an allosteric effect. This can activate the enzyme or inactivate the enzyme. And number 9 and 10 we'll see quite a bit. 9 is phosphorylation and dephosphorylation of the enzyme. As we discuss glycogen metabolism, we'll see this phosphorylation comes into play a lot with regulation. And then lastly, there can be a regulatory protein that binds to the enzyme and alters its activity. So how can the concentration of the substrate affect a reaction? If you recall when we discussed Michaelis-Menten kinetics, we discussed how this graph shows us the activity of an enzyme. So what we saw is that relatively low concentrations of the substrate, we saw a pretty steep increase in velocity, which is the speed in which the enzyme is working. So as long as we set up a situation within the cell where we're keeping our substrate concentration low, then we're going to have the enzyme able to constantly work on the substrate. We're also going to want an enzyme that has a pretty high Km. The Km can tell us a lot. In this example, the Km is 5 millimolar. So if we were anywhere in concentration under 5, then this enzyme is going to be pretty fast acting. But if we get above 5, we're going to begin to approach the maximum velocity, and then our enzyme is going to be pretty saturated with our substrate. Thinking about the phosphorylation of an enzyme to affect its activity in a little bit more detail, most of the time we're going to see the enzyme can be acted upon by a kinase. So if we have a kinase, it's going to take a phosphate from an ATP and add the phosphate to the enzyme. The phosphate's always going to be added to a amino acid that has a hydroxyl group. So you're going to see serine, threonine, and tyrosine coming into play a lot. This phosphorylated protein or enzyme can be dephosphorylated by a phosphatase. So you need to really pay attention to the kinase and phosphatase. That always tells you what's going on and that this is a enzyme that's regulated by phosphorylation. What it will look like if an enzyme is regulated by a regulatory protein, then you're going to see an enzyme that has typically a couple different subunits. So you'll have the catalytic subunit, a regulatory subunit, and then sometimes even other subunits. And you have to have them all in the correct location within the cell. They have to all be produced in the correct amounts within the cell to get the desired effect in the metabolic pathway. Two of the most common allosteric regulators of the reactions are ATP and AMP. And if you think about that, one of our big goals as a human is to make sure we have enough energy. The availability of ATP drives a very large majority of our reactions. But what may be a little bit surprising to you is the concentration of AMP is actually much more important than the concentration of the ATP. So if we look at this picture, you can see whenever we're talking about AMP regulation, the presence of AMP is going to activate fatty acid oxidation, glucose uptake, glycolysis, make you want to eat. It's going to make your skeletal muscle function specific ways. Now, AMP can be a negative allosteric regulator with some other things, but AMP is one of the most important regulators within our different pathways. So that's enough discussion just using generalities. Let's go ahead and move into thinking about how our glycolysis and gluconeogenesis pathways are regulated. Now, not every enzyme is going to be regulated. The main enzymes we're not going to regulate are the enzymes that we share within these opposing metabolic pathways. We regulate the reactions that have a high negative delta G. They're going to be the reactions that have different enzymes. So if you think about it, why do we want to do that? Why do we want to regulate the steps that have different enzymes? It allows us to have much more control of what's going on within our pathways. So our three steps that are going to be really paid attention to are the activity of hexokinase, the activity of phosphofructokinase, as well as the activity of the pyruvate kinase. Experimentally, they have actually determined that the activity of hexokinase is more important than the activity of any of the rest of the enzymes when we're thinking about regulation. So what they've done is they took a closed system and they increased the amount of individual enzymes. And what they observed was the glycolytic flux, which is the amount of glycolysis, was affected most when we increased the activity of hexokinase. So hexokinase is actually the most important enzyme that's regulated. You may recall when we began our discussions, I mentioned that hexokinase exists in multiple isoforms. There are actually 4. Hexokinase 1 is expressed in almost all tissues, but just at different levels. Hexokinase 4, sometimes called glucokinase, is expressed only in the liver. And then in the muscle, we typically use hexokinase 2. But just for simplicity, let's look at the difference between hexokinase 1 and hexokinase 4. If you look on the graph, you can see hexokinase 1 that is found in almost all tissues. It has a relatively high Km, a relatively low Km, because it approaches and reaches its maximum velocity very fast. But if you look at the glucokinase, hexokinase 4, it has a much higher Km. It has a higher concentration of glucose before we get to half of maximum velocity. This allows hexokinase 4 in the liver to continue functioning even when you have a high amount of glucose within the liver. The hexokinase activity will stop sooner in other tissues. Hexokinase 4 is also not inhibited by glucose 6-phosphate, which is the product of the hexokinase reaction. So this hexokinase 4 within the liver is going to continue to work much longer under much higher glucose and glucose 6-phosphate concentrations than any of the other hexokinases. In addition, the hexokinase we can make some other things happen by binding a regulatory protein so that the liver is always capable of not just using the glucose, but always transporting the glucose into the cell. So what we'll do is when we're in a condition that has very low glucose concentrations, such as a fast, okay? So you have low glucose concentrations. What you're going to do is take this enzyme, hexokinase, move it into the nucleus so that it's bound to a regulatory protein, so that hexokinase is finally going to stop working. So if you think about it, we have to have some sort of way of regulating this, because even though the main job of the liver all the time is to utilize the glucose, so we always want it to function, there are some times when we need it to stop, and that's why we use this regulatory protein to do so. So now looking at the regulation of phosphofructokinase, which is the step where fructose 6-phosphate is converted into fructose 1,6-phosphate. If you recall, we called this the first committed step of glycolysis. The most simplistic regulation that occurs here is that this enzyme is inhibited by ATP and activated by AMP and ADP. The inhibition by ATP tells us that we know in order for when this step begins to occur, we're pushing towards ultimately producing more ATP because we're going to make pyruvate that can move into the citric acid cycle. So if we don't need ATP, if we have a lot of ATP present, we don't need any more, then we don't need to go through this step of glycolysis. So ATP inhibits. Well, if we're really low on ATP, that means we're going to have a lot of AMP and ADP. We need this reaction to go. So AMP and ADP are going to activate the reaction. Now, citrate also inhibits this reaction. Citrate is considered kind of a major junction. Whenever you have high concentrations of citrate, that means you've already probably made the amino acids you need. You've made the fatty acids that you need. You probably don't need to go through your pyruvate oxidation into the citric acid cycle anymore. So the citrate's just going to go all the way back and stop glycolysis as early as possible. Now this fructose 2,6 bisphosphate, that is probably the most challenging to understand of all the regulations, and so that one's coming. So let's go ahead and compare the activity of phosphofructokinase to fructose 1,6 bisphosphatase. So just to orient you, if we're going in the direction of fructose 6-phosphate to fructose 1,6 bisphosphate, this is the direction of the phosphofructokinase. So same thing we saw on the slide before, activated by ADP and AMP, inhibited by ATP and citrate. The opposite direction, the movement in gluconeogenesis, the fructose 1,6 bisphosphatase activity, is inhibited by AMP. So the opposing reactions have opposite regulation, where one direction is activated by AMP, the other direction is inhibited by AMP. So now let's look at the activity of that fructose 2,6 bisphosphate. Now, fructose 2,6 bisphosphate, it's not part of glycolysis. It is simply a regulator. It is produced only to have an effect on the phosphofructokinase and the fructose 1,6 bisphosphatase. Okay? So just in general, the presence of the 2,6 bisphosphate is going to activate glycolysis, where the presence of the 2,6 bisphosphate is going to inhibit. So here we go. This is how the fructose 2,6 bisphosphate regulation works. Okay, so let's start over here. In the event that we have glucagon present, glucagon tells us that we have a low concentration of blood glucose. That is telling the liver, I don't have any glucose in my blood. I need you to release some. So what we need to do is stimulate gluconeogenesis. The presence of the glucagon is going to activate a kinase, phosphorylate fructose 2,6 bisphosphatase. So now it's active. The active fructose 2,6 bisphosphatase is going to stimulate gluconeogenesis because we're breaking down the fructose 2,6 bisphosphate. I kind of know, wow, that's one of those you're going to have to say several times for it to work. If we look in the opposite direction, the presence of insulin. Insulin tells you you have high blood glucose. So you need the liver to take all that glucose in, use it as fuel if needed, or to, more importantly, make some glycogen. Okay? So the insulin activates a phosphatase, which causes the fructose 2,6 bisphosphatase to become inactive, and now we have phosphofructokinase 2 activity. This activity is going to stimulate or increase the presence of fructose 2,6 bisphosphate. The presence of fructose 2,6 bisphosphate stimulates glycolysis and inhibits gluconeogenesis. So going back through it one more time, just because it's a little bit confusing, okay? So we're talking about this fructose 2,6-bisphosphate. It is produced to be a regulator of glycolysis and gluconeogenesis. So you see this PFK2, that's what makes fructose 2,6-bisphosphate. So when the PFK2 is active, it makes more fructose 2,6-bisphosphate. We stimulate glycolysis, inhibit gluconeogenesis. The FB, the fructose 2,6-bisphosphatase, 2, is going to break down all that fructose 2,6-bisphosphate. So when we have an active fructose 2,6-bisphosphatase 2, due to phosphorylation, we're decreasing the amount of the fructose 2,6-bisphosphate, inhibiting glycolysis, and stimulating gluconeogenesis. So if we want to think about this a little bit, maybe push a little bit harder, think about the question, why when we eat a diet that's very high in sugars, will we end up making and storing more fat? So the high amount of sugar is going to cause the release of insulin. The presence of insulin activates and makes the PFK2 active. PFK2 makes fructose 2,6-bisphosphate. The fructose 2,6-bisphosphate stimulates glycolysis. Glycolysis continues, activating the citric acid cycle, producing high amounts of acetyl-CoA. Now, we're not there yet, but what's going to happen is when you have high amounts of acetyl-CoA, that's going to lead to an increase in fatty acid production. Now let's look at the regulation of pyruvate kinase. Pyruvate kinase is going to be regulated by the presence of fructose 1,6-bisphosphate. Now that is not the substrate of the enzyme, but if you've got a lot of fructose 1,6-bisphosphate, that means glycolysis needs to keep going. So we're going to activate our pyruvate kinase. Pyruvate kinase is going to be inhibited by ATP as well as any other signs that we have plenty of energy. It's going to be inactivated by signs of glucose depletion, which is going to mainly be your glucagon effect. But glucagon doesn't affect all organs. We're thinking mainly about the liver. So the lower the sugar, that's going to make your pyruvate kinase inactive. That way all the sugar is going to be sent to your vital organs. So the next slide shows you the regulation of the pyruvate kinase. Okay. So over here you can see whenever the pyruvate kinase is phosphorylated, it's inactive. That happens in the presence of glucagon. When the pyruvate kinase is not phosphorylated, then it's not going to be as active. Then coming over here, looking at the effects of pyruvate kinase in all tissues. It is activated by the presence of some of the initial products. It's inhibited by the presence of ATP, as well as some of the other ultimate products if you go through many different steps of glycolysis. So just to review, we need to think back to the fact that when we have pyruvate, we can go in multiple directions. So pyruvate can serve as the new source of glucose, and that's kind of where we're going with this. We can take the pyruvate, convert it into glucose, and make glycogen from it. Or that pyruvate, as we continue to discuss, it can eventually become acetyl-CoA, enter the citric acid cycle, and make energy.